Hello and welcome student. Today we'll learn a structure of typical ovule. Now before that we'll see what ovule is and we'll understand where ovule is present. Now a typical flower, it has male and a female reproductive unit. Now in this female reproductive unit, it is called as a gynoecium. Now this gynoecium, it is nothing but a whorl of individual members, that is carpel. Now this carpel, it has three parts, that is stigma, style and ovary. Now this is a single carpel. Here we can see this is stigma, this is style and this is ovary, and inside this ovary a structure female gamete is present, and that is nothing but ovule. Now this style, it can be hollow style either, or it can be solid style from inside. Stigma, it is such a part where pollen grains after pollination come and settle on this stigma, and onto this stigma it forms a pollen tube, which we say a male gametophyte. Now that pollen tube or male gametophyte pollen tube travels from this style. So it is hollow from inside, or it can be also solid, that is filled with the cells. Now this stigma, it has many functions, like one of it is it secretes a stigmatic secretion, because of which this pollen, it forms pollen tube, and that pollen tube, it has male gamete, and that male gamete through that pollen tube travels till this ovule, and inside ovule we have egg, which is a female gamete, and their fertilization occurs. Now we are focusing on this ovule. Now we'll zoom this part and we'll see the structure, and we'll understand each layer and its function of each layer of this ovule. Now students, here we have the structure of a typical mature anatropous ovule. When we say anatropous, it means this opening or this micropyle end, it is towards downside. When this opening or micropyle end is towards upper side, then it is called as a orthotropous ovule. Based on this micropyle opening, where it is, based on that different types or kinds of ovules are there. We'll go in detail of it in some other video. Now this structure of ovule we'll understand. So we can divide this structure of ovule into two parts. Now that two parts are nothing but this is body of ovule, and this much part we say it is the stalk of ovule. So we'll divide into a stalk and another it is body. Now we'll see what is there in this stalk. Now, and this is stalk of ovule, and this we say it is funiculus. So this funicle, the ovule, it is attached to the placenta of the ovary by a stalk, and that stalk we say it is funicle. So this part is a funicle. Now the point of attachment of body of this ovule with the funicle, and that point of attachment we say it is hilum. So that part we say it is hilum. Now we will see some other part. So in a stalk we have a funicle and the point of attachment, that we say it is hilum. Now another part that we say it is a raphe. So here in this diagram we can see this raphe. Raphe, it is nothing but this funicle continues beyond the hilum, along the body of the ovule, and that ridge we say it is a raphe. Now this body, will focus on this body. Now this body of the ovule, it consists of inside this much part that we can see it is in the purple color, and that part it is nothing but nucellus. So the body, it is having parts like the very first part is nucellus, another part we will write is integument and micropyle, and we'll see one by one these parts. Micropyle. Okay, so first one, that is the nucellus. So what is nucellus? It is the main central parenchymatous cells are present surrounding this embryo sac. So this central part we say this is embryo sac, and beyond or surrounding this embryo sac the parenchymatous cells are present, and that we say nucellus. So what is the function of this nucellus? It is megasporangium, and here megasporogenesis takes place. Now the another part is integument. Here we can see these covering of this ovule. These are nothing but integuments. Now this integument, it surrounds this nucellus, and which develop from this chalaza. This is one end, chalaza surround it completely except the small portion. So integument, it is leaving this much space, and the function of this integument, obviously as it is from the outer side, so its function is protection to this nucellus and to this embryo sac. And later this integument, after fertilization, it is going to convert into a seed coat. So whenever you have seen some seed, it is having seed coat. So that seed coat is nothing but this integument. Now that integument, it is having the outer integument and the inner integument. So outer one, it is called as the testa, and the inner one, it is called as a tegmen. Now understand student, this ovule, it is such a part which is after fertilization, it is going to convert into seed, and this integument, it is going to convert into seed coat. Now we'll see the next part, that is micropyle. So here very clearly we can see this is the small gap left at integument. It means this integument leave narrow opening, and that opening, it is called as a micropyle. Now why it is called opening? Because whatever the pollen grains are there, when it settles on the stigma, it forms a pollen tube, and that pollen tube, it has male gamete, and it has to transfer till this egg cell. Now this pollen tube, it enters from this micropyle. So it is, we can say, it is the passage for entry of pollen tube inside the ovule, and during seed germination it also allows entry of water and radicals to come out. It means this micropyle, after fertilization, when this ovule is going to convert into a seed, at that time that seed, when we sow that seed, at that time it germinates, and while germination, this micropyle or this opening, it allows the entry of water to come inside, so that seed, it will grow. And even radical, which is the very first part when from which the root arises, and that radical also, it comes out through this opening. So that is also one of the important part that we say it is micropyle. So we have understand this is the stalk, that is funicle, the point of attachment, that we say it is a hilum. Then we have this ridge, and that ridge we say it is raphe. These covering, two coverings, that we say integument, its function is protection. This parenchymatous cells which is surrounding the embryo sac, it is called as nucellus, and this, the opposite to the micropyle, the whatever end is there, that end we say it is chalaza. Now we'll see what is inside this embryo sac. Now we'll focus on this embryo sac, what is present inside the embryo sac. So see here student, we'll focus on embryo sac. Embryo sac, it is large oval cell which is embedded in nucellus towards the micropyle end. It is known as the embryo sac. So I have drawn here embryo sac. Here this cell which is present inside that nucellus. This is called as a embryo sac. Now we'll see what are the parts of this embryo sac. It is very important thing we have to remember is it is a 7-celled and 8-nucleated structure. It means that this embryo sac, it is having 7 cell and it is having 8 nuclei. So because of that it is called so. Now it has 3 parts, that is egg apparatus, second is a central secondary nucleus, and third is antipodal cells. Now with the very first part we'll understand egg apparatus. What is egg apparatus? Egg apparatus, it consists of 2 synergids and 1 egg cell at a micropyle end. Now here we can see this is egg apparatus and it is having this central egg and these 2 synergids, 2 cells. So how many cells are there? There are 3 cells at this micropylar end, and the central one it is called as egg, and another is 2 cells, these are called as a synergids, and together we say it is egg apparatus. Now this we'll see what synergids are. Synergids has prominent structure. It is known as that prominent structure. It is known as a filiform apparatus. Now, students, this filiform apparatus, it has one function, that is attraction of that pollen tube which I mentioned. So that pollen tube attracting that pollen tube, it is the function of this filiform apparatus. So synergids, these are also very important part of this embryo sac. It means it guide the path of pollen tube towards egg. It means attraction of that pollen tube. And it also acts as a shock absorber while penetration. It means when that pollen tube enters inside this embryo sac, at that time this synergids, these get degenerated afterwards, and it functions as a shock absorber, and also it functions as a nourishment. It means it provides the nourishment to that further embryo. Now another part, that is the center cell, and it is very important cell, and that is nothing but egg cell. This is what a female gamete is. Now here only, means with this egg cell, that pollen, or we can say male gamete fuses with this egg cell, and that we say it is the first fertilization, which we'll see in fertilization part. Now this egg, it contain female gamete. Male gamete fuse with this female nucleus. Now its function is it converts into a diploid zygote. It means after fertilization, this egg cell or this fusion, it give rise to zygote. So very important part is zygote. It is forming from this egg cell, which we say it is the first fertilization. Now we'll see what is this central cell. In the center of this cell, it is having 2 polar nuclei. It means these are 2 haploid nuclei N, and it fuses together and it forms the secondary nucleus. That means this central cell, it is diploid in nature. This is very important, student. This egg cell, it is haploid, and this central cell, it is diploid. Now these 2 cells or these 2 nuclei are very important, because with this nuclei only the fertilization of that male gametes occur. Now we'll see what antipodal cells are there. Antipodal cells, these are present, we know this are present at chalazal end, and their protoplasm, it is rich in starch, protein, accessory cells, enzymes, etc. And a very important thing is these are accessory cells. It means it helping for this all process, and these antipodal cells, these degenerate later. It means it is not no more. And function of this antipodal cell, it limits chalazal expression. So we have seen what is there inside this embryo sac. We have seen antipodal cell, central cell, which is a diploid secondary nucleus we say, and this is central egg cell, and 2 synergid. It means 1, 2, 3 cells here, 3 cells here. So we have a 6, and the central 7 celled 8 nucleated structure. Now for your reference and for your note purpose, you can have these notes also. Here I have mentioned all the parts inside this ovule. So ovule, once again we'll revise very shortly or very quickly. The integumented megasporangium, ovule, it is nothing but megasporangium of seed bearing plants, and this converts into a seed after fertilization. Funicle, the ovule is attached to the placenta of the ovary by this stalk, and that stalk, it is nothing but funicle. Hilum, it is the point of attachment. Raphe, it is nothing but this funicle continues beyond the hilum, and that forming a ridge, and that ridge is nothing but raphe. Now that body it is, we are dividing this body into nucellus, integument and micropyle. Nucellus, it is central parenchymatous cells, and it is only the megasporangium where the megasporogenesis takes place. Now integument, it is surrounding this nucellus, the covering is present, and that we say integument. Now this integument, on the basis of this the number of integuments, there is terminologies like, if it is a single integument, then we say it is unitegmic, and if there are two integuments, then we say it is bitegmic. Unitegmic example is it is present in most of the dicotyledons, and bitegmic it can be present in a dicots and monocots. Now if it is collar-like outgrowth, then it is called as aril, and if these, in some parasites like viscum or these integuments, there is no integument. If it is integuments are absent, then it is called as a ategmic. So based on this, there are different terminologies. So shortly I have given you these terminologies. Now micropyle, it is the small gap which is left because of this integument, and function of that micropyle, it is nothing but passage for entry of pollen tube. So I hope, student, this all structure of ovule, it is clear to you. Based on this number of gynoecium, there are different fusions also present, so that part we will cover in some other video. So structure of ovule is clear to you. If there any doubts are there, then you can comment. Thank you so much.